Welcome, everyone. Today, we are diving into a critical topic in medical entomology known as host specificity. If you have ever wondered why some mosquitoes seem to hunt humans relentlessly while others completely ignore us, you are in the right place. This single biological trait is the key to understanding how deadly diseases spread through ecosystems, and more importantly, how we can stop them. Let us get started. To understand what host specificity is, imagine you have a friend who is an incredibly picky eater. If their favorite brand of pizza is not available, they would rather go hungry than eat anything else. In the fascinating world of insect vectors, many insects behave exactly like this picky friend when it comes to choosing whose blood they want to drink. This biological preference is what we call host specificity. It matters immensely because it helps us understand exactly why certain insects spread diseases to humans, while others pose absolutely no threat to us at all. Now that we understand the basic idea, let us look at how we formally classify these behaviors. Host specificity is defined as the degree to which a vector restricts its feeding to a limited group of hosts. We use three main categories. When an insect feeds exclusively on one single type of animal, we call it monophagous. It is highly inflexible. If it feeds on a few related species, it is classified as oligophagous. Finally, if the insect is not picky at all and feeds on many different animals, it is known as polyphagous. These classifications are our first step in predicting insect behavior. Building on those broad classifications, medical entomology uses very specific terms when human health is on the line. We classify insects based on their ultimate target. Insects that strongly prefer human blood are called anthropophilic. On the other hand, insects that prefer animal blood are known as zoophilic. Understanding whether a local mosquito population is anthropophilic or zoophilic allows scientists to predict which insects are capable of acting as biological bridges. This tells us exactly how diseases might jump between animal reservoirs and human populations. Why do we care so much about the dietary preferences of a tiny mosquito? Because this preference directly controls the pathways of infectious diseases in the real world. If a mosquito is strictly zoophilic and only bites birds, it will never transmit avian diseases to humans. It acts as a natural dead end. However, if an insect has low host specificity and bites both infected animals and healthy humans, it becomes a highly dangerous bridge vector. These bridge vectors are exactly what trigger massive zoonotic disease outbreaks. Without this knowledge, public health officials would waste millions spraying chemicals on harmless insects. To move beyond simple observation, entomologists need to scientifically measure these preferences. This brings us to a mathematical tool called the forage ratio. We cannot just count how many humans a mosquito bites, because there might simply be more humans around. We must compare the actual diet to the availability of hosts. In this beautifully simple formula, W sub I represents the forage ratio. The variable O sub I is the observed proportion of blood meals from that specific host. Finally, P sub I is the proportion of that host actually available in the environment. If the ratio is exactly 1, the insect bites randomly. If it is much greater than 1, the insect actively hunts that host. If it approaches 0, the insect actively avoids it. Let us see this formula in action with a concrete example. A research team is studying a village with 800 humans and 200 cows. They test 100 mosquito blood meals and find 40 came from humans. We need to find the forage ratio for humans. First, we calculate the environmental availability. 800 humans out of 1,000 total hosts gives a proportion of 0.8. Next, we find the observed diet. 40 human meals out of 100 total gives 0.4. Finally, we divide the observed proportion by the available proportion. 0.4 divided by 0.8 gives us a forage ratio of 0.5. Since this is less than 1, it physically means the mosquito is actively avoiding humans. Now that we have calculated the ratio for humans, let us repeat the exact same process for the cows in the village. The village has 200 cows out of 1,000 total hosts, so the availability proportion is 0.2. Out of the 100 blood meals tested, 60 came from cows, giving an observed proportion of 0.6. We apply our forage ratio formula by dividing the observed 0.6 by the available 0.2. This gives us a final forage ratio of 3.0. Because this value is much greater than 1, it physically means the mosquito has a very strong host specificity for cows. It is actively seeking them out over the humans. While the forage ratio is great for general ecology, public health workers dealing with diseases like malaria rely on another vital metric. This is the human blood index. This index tells us the exact probability that a particular insect vector has fed on a human being rather than an animal. In this formula, H represents the human blood index. The variable N sub H is the number of blood meals specifically identified as human blood. The variable N sub T is the total number of blood meals tested from the captured population. When the number of human meals increases, the overall index approaches 1. A high value is a massive warning sign, indicating that the disease transmission risk to the human population is extremely high. Let us apply the human blood index formula to a real outbreak scenario. Health workers trap 500 mosquitoes. Tests show 350 contain human blood, 100 contain dog blood, and 50 are completely empty. We need to calculate the transmission risk. Our first crucial step is to find the total number of actual blood meals. We must completely ignore the 50 empty mosquitoes because they provide no feeding statistics. Adding the human and dog meals gives us a total of 450. We then divide the 350 human meals by this total. The result is approximately 0.777. This physically means roughly 78% of the feeding mosquitoes are biting humans, explaining why the malaria outbreak is so severe. Let us bring all these mathematical concepts together visually. On the screen, you see a schematic representing the exact scenario we just calculated. On the left is our insect vector, the mosquito. Notice the two pathways it can take. The dashed arrow pointing toward the human figure represents a forage ratio of less than 1. The thin dashed line visually reinforces that this is a weak preference or active avoidance. Conversely, the thick, solid black arrow pointing toward the cow represents a forage ratio greater than 1. This clearly shows a strong zoophilic preference. Diagrams like this help epidemiologists quickly map out how diseases will flow through a local ecosystem based on the math. We have covered a lot of ground today, so let us summarize the most important ideas. First, host specificity is the biological compass that dictates what an insect vector chooses to bite. We learned that anthropophilic vectors target humans, while zoophilic vectors target animals. Second, we explored how to quantify this behavior using the forage ratio, which compares an insect's actual diet to the availability of hosts in its environment. Finally, we calculated the human blood index to measure the direct probability of human feeding. By mastering these mathematical tools, you are now equipped to understand the very first step in designing targeted strategies to protect human populations from deadly vector-borne diseases. Great work today!